With a major selection of the looks your kiddos love starting at just $4 in the Walmart app, a new school year means a stylish new them. Who knew? Find skater skirts and Crocs that say, I'm casual but cool. Sparkly sneakers that say, it's my classroom, you're just learning in it. And preppy polos that say, I'm the teacher's pet, even if they're mommy's menace at home. Find their favorite styles starting at just $4 in the app. The Walmart you thought you knew is now new.
The microbiome has become a bit of a buzzword in the space of personal wellness. But what does it actually mean? And what is the difference between the microbiome and gut health itself? And most interestingly, what can we learn about the human microbiome from the microbiome of domesticated animals such as dogs and chickens? Bushra Schuitemaker is a microbiologist specialising in the microbiome of domestic animals. Having previously worked in labs at the Wellcome Sanger Institute, Cancer Research UK and the Quadram Institute, Bushra is now the Head of Science and Research at Biome9, a personalised data-led brand looking to revolutionise the pet wellness space using microbiology and AI. Bushra, thank you for joining me on the podcast. To start with, could you tell us a bit about how your interest in the microbiome of domestic animals came about?
Yes, of course. And thank you so much for having me. So I did my undergraduate in zoology. I've always been obsessed with animals, and I think domestic animals have always held a special place in my heart. I grew up not being really allowed to have any pets. Eventually, I wore my mum down and we had a pet cat. And then at one point, I also had a small group of chickens that I incubated and hatched on my kitchen counter. So I think domestic animals have really, I've always had like a really deep love for them. And even with those chickens, my mum was very against me having chickens. But one day I just came home from high school with an incubator I borrowed from a farming friend and 6 eggs that were all fertilized and just set it up before she came home from work. So there wasn't much that she could do about it once the process has started.
Love that.
So I've always found ways to have domestic animals in my life, even if they weren't directly my own. And after my undergraduate in zoology, I actually started working for the Wellcome Trust Sanger Institute and also Cancer Research UK. And with this job, I actually moved away from animals. I was looking more at cancer as a disease and looking for immuno-oncology drug targets, particularly for metastasis, so that process of cancer spreading around the body, because it's very difficult to find therapeutics for that. And although I was really interested in the experimental side and the science of it all, and I've always loved biology, for me, everything always came back to, but how does this apply to animals? And I think cancer is one of those diseases that is so widespread, not just through humans and cancer being, a group of so many different diseases, but also how widespread it is across the animal kingdom. And what really fascinated me is looking how far back it went. So the fact that they actually find evidence of metastases in dinosaurs' remains shows how much of an old process and disease cancer is and how it affects so much more than just the human population. And then there are also instances of cancer that only affect specific species, such as the Tasmanian devils and the facial tumours that almost completely wiped out their species. So I was really interested in what of those biological processes are shared, and is there anything that we can learn from the animal kingdom that could help benefit human medicine? Similarly, are there things that we're doing in human medicine that could be helping animals? And so that's when I really learned about the concept of one health and one medicine. So the idea that we shouldn't be working with humans and animals and the environment separately, but this is all part of one larger health system. And so I ended up publishing a couple of papers that was looking at comparative oncology. So these shared features in cancer and oncology between animals and between humans, looking for those shared benefits. And one of the other things that is incredibly diverse and shared across the animal kingdom is the microbiome. And what's so fascinating about the microbiome is it's made of a community of microorganisms. And these microorganisms have existed long before any mammals did. Cyanobacteria existed 2.4 billion years ago. So there's this incredible ecosystem of microorganisms that is living inside and on every other mammal, bird, reptile, fish that exists in the world. And again, what really fascinates me is those shared processes. What microorganisms are shared between us? What microorganisms have a similar effect across these different hosts? So I did my PhD in looking at poultry biomarkers. My work at Cancer Research was really focused on those drug targets, and it translated really easily to that biomarker discovery for poultry. And with poultry, obviously, collecting a microbiome sample basically just involves me collecting a poo sample from a farmer, which is very easy to do. And I'm not stressing the animal out in order to collect it. So it's one of the reasons fecal testing is so good, especially when we're thinking about enhancing animal welfare and better ways of monitoring really important animal populations, such as chickens. One of my favorite chicken facts is that there are 4 chickens for every human on the planet. And as someone who loves animals, has my own personal history with chickens, I couldn't imagine anything better than helping one of the most dominant animals on the planet by finding new ways of monitoring their health and welfare, but just using those poo samples and identifying those different microorganisms.
Most of the time, when we're hearing about the microbiome, we're talking about humans and what's going on in our gut. But how similar or different is our microbiome to the species around us?
Well, especially domestic animals, because they spend so much time with humans, there's almost like a humanization of their microbiomes that occurs. So even with chickens, when they actually look at the microbiome of chickens on a commercial farm, and they look at the microbiome of the humans working on the farm, they find similar or shared species of bacteria, and that's because of that direct interaction. So a lot of the microbiome comes from things like our diet, but there's a huge amount of it that comes from our environmental exposures. I like to remind people that everything has a microbiome. Surfaces on the table have a microbiome. The chair you're sitting on public transport has a microbiome. And having all of those different environmental exposures during our life is help what's to create that ecosystem of microorganisms. And it's the same for animals as well. So when domestic animals are sharing the same space as us, whether that be on a farm, whether that be our pets in our home, they're exposed to similar environmental microbes as what we are. And so we end up sharing a lot of them. What's really interesting about particularly pets and specifically dogs is that because they have evolved alongside humans for tens of thousands of years, it means that they are even more similar to the human microbiome than lots of other animals. Because dogs have evolved to live in our environment and to eat very similar foods to us, we see a lot of similarities shared between that human microbiome and between the dog microbiome as well.
The environment obviously plays a really significant part in developing the microbiome, but are there pre-existing processes that also have a role in shaping it?
Yeah, so one of the questions I will often get asked is, where does the microbiome initially come from? And I think when we look at the animal kingdom, we can look at 3 really interesting examples just looking at mammals. So if we take a dingo, for example, so Canis lupus dingoes, they are a wild dog that lives typically in Australia, and they are placental mammals, so they give birth vaginally. Their initial microbiome comes from that vaginal delivery, but also the lactation period. So, that time that the pup will be spending with its mum and getting all of those microbes as well as nutrients from her milk. Now, if we look at another mammal, say a platypus, which is a monotreme, they lay eggs. So their young are not getting the initial microbiome coming through that vaginal birth. Instead, they're getting it from the shell of the egg. Now, platypus, just like birds, have a cloaca. So that's a single urogenital orifice that does everything. So when the platypus are laying their eggs, on the outside of those shells is some fecal microbiome. So when those platypus babies, puggles they're called, are first hatching, they get their initial microbiome from the outside of the shell of the egg. And that's the same across all animals that are born via hatching and via eggs. So it's the same for our chickens as well. Something that's been really interesting that they do, especially in the USA and Canada, is they've actually created a microbiome spray from the adult chicken's feces. And they spray this on the eggs in commercial hatcheries so that when the chicks are hatching, they're getting a copy of what it would be if they were actually hatching in a nest with their mother. So they're still getting those initial microbes when they're pipping through the egg. And then another mammal to consider as well is, of course, marsupials. So we think of kangaroos, they give birth to a very, very underdeveloped small embryo that spends part of its growing period, a significant part, in that pouch of its mother. So again, thinking of those different environmental exposures it would be getting whilst in the pouch, as well as what it's getting from the mother during that lactation period. So those are the things that really have an impact early on in life. And we know early on in life, for all animals, has a lasting impact on the gut microbiome. In dogs, and we also see this in humans as well, when there's really, really early exposure to antibiotics, which are an essential life-saving treatment, but that can actually have really long-lasting effects on the microbiome that can be seen years later. And so we really know that that initial microbiome has a really big effect in sort of starting up that long-lasting gut ecosystem.
You can see across all sorts of different species that processes have developed to allow the microbiome to form, which we'll get into later. But the existence of these processes alone just shows how important the microbiome is. So, what is it actually for?
I mentioned that we have microbiomes all over our body. I take a lot of focus on the gut microbiome, and that's the one that is actually living in our gastrointestinal tract. But it's important to remember we also have a skin microbiome, an ear microbiome, a mouth microbiome, and they're all distinct throughout our body. But that gut microbiome is what's really, really key. Sometimes it's described as the hidden organ because we didn't realize how important it was until only really maybe a decade ago. That microbiome in the gut helps with digestion, being one of its key things that it does. So the body only partially responsible for digestion, and the further down the gastrointestinal tract food travels, the more it's depending on the gut microbiome, so the bacteria and fungus and viruses living in that gut ecosystem, to help further break down that food into really helpful molecules called metabolites that can be used across the entire body. So the bacteria helps to ferment the food. This creates things like short chain fatty acids, sugars, alcohols that can be used by the actual cells of the body itself. So it really helps to turn your food into things that can be used across the whole body. So when people say you are what you eat, we're really starting to understand how much the bacteria have a role to play in that. But digestion is just one part of it. The reason why that microbiome is so important in that early development, and we see that across the animal kingdom, is because it helps to stimulate the immune system. That's what's so important about the microbiome in early life, is it really helps to develop a strong immune system. And a lot of the immune system have pathways that are shared with the microbiome. So even throughout our life, there is an interaction between the immune system and the gut microbiome. We've also discovered loads of other axes across the body where the microbiome has a direct influence. One of the very exciting ones that a lot of people are talking about is the gut-brain axis. So the ability of gut bacteria and the brain to have an influence on each other. And this is a bidirectional pathway, partly facilitated by the vagus nerve. So the thing that actually connects your gastrointestinal tract to your brain and to your nervous system. And it makes a lot of sense when we think about things like gut feelings and how when we are stressed out, a lot of people will experience a really upset stomach. And so there is that bidirectional pathway of stress can cause issues within the gut microbiome, but also an upset gut microbiome known as dysbiosis. So when the gut is no longer balanced, that can also exacerbate issues associated to, say, anxiety and mental health concerns. What's really interesting about studying across animals is we can start to look at this in dog populations. We know that there are dogs that do suffer with reactivity and anxiety and aggression. And based on what we know from the human studies of that gut-brain axis, we can see if any of those processes are shared in dogs and see whether or not the microbiome is actually a useful option in terms of managing and treating these conditions.
We can learn a lot about humans from domesticated animals thanks to that massive overlap in our microbiomes. But can we also learn from domesticated animals about other animals and how that domestication has occurred or how they differ from their wild animal counterparts?
One of the really interesting things when it comes to looking at the human genome and also the canine genome is the increase in a really specific gene. So this is the AMY2B gene. It's an amylase gene that helps to facilitate starch digestion. Humans have about 20 copies of this gene. Wolves and also dingoes only have 1 copy of this gene. Domesticated dogs, just like humans, have 20 copies of that gene. So that goes to show the type of co-evolution that has occurred on a genetic level in order for domestic dogs to be able to digest foods such as starches, which are very common in human diets. Now, unfortunately, time travel is not available. And so we can only make theories about actual dog evolution because we can't go back and observe it. But we make these theories and then we find the evidence that helps to either validate or disprove those theories. One of the theories looking at dog domestication thinks about that first interaction between wolves and between humans. So there's a theory that wolves started living closer to humans because humans would leave a lot of mess behind, scraps of food, and this was beneficial to the wolves who would be able to get an easy meal if they were living really close to humans. There is also the theory that wolves helped humans in terms of hunting, and so that relationship started to build. Because dogs were very likely to be eating human food, what we don't know is whether or not that increase in that AMY2B gene happened as part of the microbiome changes adapting in order to eat that human food. And it's a bit of a chicken and egg situation.
Yeah.
Did the domesticated dogs develop that genetic variation first, or did the microbiome change first, which therefore drove this genetic variation that we see within the domestic dog that allows them to have that starch digestion? And we've got a really interesting opportunity now looking at the difference of urban and countryside foxes. There's some fascinating research occurring at the University of East Anglia that's looking at this evolution of particularly urban foxes. And they've found that urban foxes are physically, morphologically changing and are different to those countryside foxes. And this could be partly to do with the fact that they've got different evolutionary pressures living in that urban environment. But the foxes have also been observed to be copying some of the behaviours of domestic animals, such as cats and dogs, in order to get that food off of humans. There's another theory in evolution, which is basically survival of the friendliest, and whether or not that also had a part to play in that domestication process. But now that we're seeing foxes are eating more of these human scraps and human foods in urban areas, one of the opportunities is to actually look at that AMY2B gene and see whether or not we see an increase of that gene alongside a change in those urban fox microbiome populations.
So it's an interesting time to hopefully be getting a bit more of an insight into that process that could have happened with dogs by looking at foxes in real time, I guess.
Exactly. It's very, very exciting. And I think being able to see those kind of what is shared between these different animals really helps to give further evidence to support specific theories.
This episode is brought to you by San Pellegrino Ciao, a new kind of flavor sparkling water. The kind made with real fruit juice, a pinch of Sicilian salt, and the sparkle of the Italian sun. With no added sugar and just 10 or less calories per can, enjoy. With love, from Italy. By San Pellegrino Ciao now.
This episode is brought to you by Indeed. When your computer breaks, you don't wait for it to magically start working again. You fix the problem. So why wait to hire the people your company desperately needs? Use Indeed's sponsored jobs to hire top talent fast. And even better, you only pay for results. There's no need to wait. Speed up your hiring with a $75 sponsored job credit at Indeed.com/podcast. Terms and conditions apply.
So, as you said, we can infer that these processes have been happening for an extremely long time. But you also mentioned that obviously everything has a microbiome, including plants and everything on Earth, basically. But what's the earliest we can kind of track that back and see examples of that in nature?
So I mentioned about the fact that the cyanobacteria evolved at least 2.4 billion years ago. And so microorganisms have existed so much longer than any animal on the planet. But I think one of the really excellent pieces of evidence that show the effect that microbes have is actually looking at soil microbiomes. Soil microbiomes are really important in agriculture. Having specific bacteria that are known to help improve the nutritional qualities of soil has been a really interesting area of research. But one of my favorite kind of soil microbiome facts are the volcano microbiomes. So after a volcanic eruption, it's actually the bacteria in the soil that have evolved to live in these highly acidic environments. They actually help to break down that volcanic rock and also create this nutrient recycling that helps kickstart primary succession after a volcanic eruption. And we can use different types of studies in biogeology to kind of look at the history of volcanic activity and the effect that's had on the geography of an area. But now we can also start to look at the biological effects. So these microbes and how they help to create an ecosystem that's stable by supporting that initial plant growth. So that's I think that's a wonderful piece of evidence of that microbiome that's shared, not just between humans and animals, but that other part of One Health is also the environment, which includes the plants and the soil. So I think that's a really great example of just how old and how established these processes are.
Yeah, I think when you think about the microbiome, volcanoes don't spring to mind. So it's a really nice way to kind of show how it is literally everywhere.
Yeah, and I always say the animal kingdom is so diverse, but so is microbiology. And one of the reasons I love biology in general is the fact that we have so many of these, not just shared processes, but even when we look at the way we classify organisms, whether or not they are animals, mammals, fish, and where they go down in those taxonomic clades, we use the exact same methods to classify bacteria and fungi because we're all part of the same larger biological system. Same as when I talk about that gut ecosystem, we use the same statistical modeling to look at an ecosystem in a woodland as what we do to look at ecosystems in that gut environment.
Now that we're kind of talking about the nitty-gritty, how you actually observe and measure these things, it's a great segue into your work at Biome 9. So how does this research and this knowledge actually get applied and put into practice?
I always think it's really important to differentiate gut health to the gut microbiome. The way that I think is a really good way to picture it is gut health is the game of football. It's the really bigger, broader term. The game of football can be gut health. But what makes up football? Well, you need to have teams of players, so they're the bacteria. You also need to have rules in place, that's the immune system. You also need to have nutrients to help feed the gut, and that comes into, say, the fans. So gut health itself is a much broader thing than just the bacteria. Gut health also has to take into consideration those environmental and host factors that we know have a long-lasting impact on the gut. It also has to take into account symptoms, medical history. These things also all contribute to that bigger gut health. What's really good to see, though, is by looking at the bacteria, which is the most dominant component of the gut microbiome, it gives quite a clear indication of when gut health is poor. So we use something called diversity. Diversity is basically a way of us being able to quantify all of the different microorganisms and also how well are they proportioned to each other. So we look at something called richness. Again, it's the same as in ecological biodiversity. It's the number of different species, just how many different species are in an environment. But then we also look at the balance. Are these species well proportioned to each other? Is it just one or two random individuals from that species, or are these more populations that are living within that ecosystem and are well established and probably a long-lasting member of that gut ecosystem? So we use this thing called diversity that takes all of those things into account in order to give us a very standardized metric to compare. And generally what we see across all animals is that higher diversity leads to better immunity and better health outcomes. So we generally associate this higher diversity to better gut health, but it is really important to look at that gut microbiome with the context of those other important factors. So the way that we do it at Biome 9 is we request a fecal sample from a dog owner. So a dog owner gets the test kit, they go out for a walk with their dog. When their dog does their business, they simply collect a small sample of it, put it into the tube, and then post it to our labs. Once they arrive in our labs, we measure out a tiny amount of the sample. We only use 200 mg, so that's only 0.2 grams of fecal sample, which is such a small amount to be able to get as much information as we do. With that fecal sample, we extract the DNA from the feces. So that's the DNA of anything that is living within that sample. So the bacteria, the fungi, the viruses, everything. We're extracting the DNA from that. We then use next generation sequencing. Because bacteria are the most abundant, they generally give us the clearest indication of what is going on. So with next generation sequencing, what we can do is look at a gene called the 16S gene that is found in every single bacteria species, but not found in any other organism on Earth. So we have a specific gene that can be found in every bacteria species, but is slightly variable between those species. So by looking at just that gene, we're able to identify all of the different bacteria species that are present in just one fecal sample. But looking at what bacteria in there is just part of the job. It's also important to understand, what does that bacteria do? What is known about bacteria? What genes does it have? And what processes is it involved with? We then combine that data with what dog owners have told us about their dog's symptoms, medical history, diet, vaccination status, when they were last wormed, when they last had antibiotics, whether or not they sleep in the bed, in their own bed, in their owner's bed, if they live with other animals. As much information as we know has an effect on the gut microbiome, we collect it. And then we have a really impressive AI platform. When I say AI, I don't mean LLM, so I don't mean like ChatGPT. This is a different type of AI, an older type of AI called machine learning. With that machine learning, we take a really healthy population of dogs and we source these dogs from working with collaborators such as the Guide Dogs Association, where we can get a really well-controlled, well-looked after population of dogs that we can monitor from puppies right into adulthood. They are controlled in the sense of giving very much the same diet, having very similar environmental exposures, and going through a very similar life as well as having regular vet checks. So by using their really healthy microbiomes, we call this our training set. So we know what a really healthy dog microbiome should look like, and then we compare that to samples that we receive from vets that have diagnosed conditions such as IBD or atopic dermatitis, really specific diagnosed skin conditions. We then look at those microbiomes and compare it to our healthy data sets in order to be able to say what bacteria might be missing from those dogs, what level of dysbiosis, that unbalancing of the gut ecosystem, has occurred, but then we also help owners to address it. For example, if the dog is really low on bacteria that are capable of degrading fiber, which helps to stimulate the immune system, we can make complementary dietary recommendations such as increasing fiber in that dog's diet. And then we can also make specific supplement recommendations too that help to really boost that recovery of the gut microbiome, especially if it is a dog that has been suffering through some kind of gastric upset, some kind of tummy unrest.
We sometimes see a link between problems with the gut and neurological problems in humans, which is a result of the gut-brain axis that you mentioned earlier. Do we see these connections in dogs as well?
Yeah, so we see that dogs that have behavioural changes, so anxiety and aggression, tend to have lower microbiome diversity. They're also missing some really key members of the core gut microbiome we expect to see in those healthy dog populations. But sometimes it's very difficult to actually decipher in a dog whether or not their behavioural issues are purely coming from the gut microbiome or if there's something more complex that's going on as well. One of the groups that we work very closely with is dogs that have epilepsy. Epilepsy is a really interesting condition when it comes to the gut microbiome. For a long time, it's been observed in humans, especially children, that changes to the diet can have a really positive effect on reducing symptoms of epilepsy. And new research has shown that actually the gut microbiome is really key in influencing the positive decrease in symptoms and severity of seizures in people who have epilepsy. And we've actually found a lot of this research is directly transferable to the canine microbiome as well. So in dogs that are suffering with seizures and epilepsy, we see a very similar microbiome as what we see in children who are also suffering with epilepsy and seizures. With those dogs, it's actually less important to increase that microbiome diversity. It's more important to ensure that they've got good balance and stability. And part of the theory as to why that gut microbiome and the brain have such a connection in epilepsy is to do with something called GABA stimulation. So it's the idea that when there is a lot of activity within the microbiome that is stimulating this GABA pathway through neurotransmitters, is exciting the neurology of someone that is therefore increasing their epilepsy severity and their symptoms as well. So we've actually been able to use that research, develop supplement plans and dietary recommendations in line with a charity called Dogaleptic that have helped us to better understand this research. And we've seen absolutely incredible results with dogs that have reduced their seizures from having them on a weekly basis to maybe once a month, just through managing the microbiome, which I think is such a wonderful thing for me as a scientist, as a dog lover to see as well. And one of the reasons that charity Dogaleptic first reached out to us, realizing there must be some kind of microbiome epilepsy association, is they personally had a dog with epilepsy and the dog had to go on antibiotics. And whilst the dog was on antibiotics, the epilepsy symptoms and seizures were really severe. And once the antibiotics stopped and the dog was started on supplements, those symptoms massively reduced again. So it's really, really important for us to better understand that. And what I'm really hoping for is, as much as we have already learned from human epilepsy and have been able to apply this to support dogs. Maybe there are things that we can learn with the dogs that have epilepsy and their microbiome, which in the future we can apply to humans as well.
AI is moving fast. So fast, it's hard to keep up. In fact, in ServiceNow's latest AI Maturity Index, scores dipped 20% from last year. But that's okay, because AI isn't a sprint, it's a marathon. You may be behind today, but tomorrow you could be a pace setter. Dive into ServiceNow's AI Maturity Index and see how you can innovate as fast as your ambitions. Visit servicenow.com/AI maturity.
What does the research landscape look like at the moment, and how are the priorities changing as our knowledge improves?
The reason why this type of microbiome research has become so big, especially in like the past decade or so, is previously we were only capturing around 5% of the microbiome. The reason for this is we were purely relying on lab-based techniques. So this meant that traditionally we would take that canine fecal sample and then we'd try and isolate live bacteria from the feces. But this meant that the bacteria, the ones that we were able to discover and observe, it was really dependent on them being able to grow under those lab conditions. And the gut is simply not lab conditions. So by using that next generation sequencing and by just looking at those specific genes, we've actually been able to see that there are so many more bacteria that we had absolutely no idea about because we weren't able to culture them. And it comes onto this idea of the matrix of knowns and unknowns, which for me is a really inspiring part of science, because I always say you don't know what you don't know. So the idea that there are still so many things that we need to discover, there are so many bacteria still that we have not discovered that are completely novel to us. So these would be unknown unknowns, things that we don't even know that we don't know yet. So I think we're going to be continuously discovering new bacteria and using these techniques that are becoming more accessible and more affordable. It is going to continue to grow exponentially in terms of our understanding of it. I think the next stage in this microbiome research is focusing on beyond the bacteria. We know that there are other microorganisms within the microbiome, the fungi, the bacteriophages, the viruses, even protists, but we don't really put as much focus onto them as we do with the bacteria because the bacteria is so dominating. But they are still important parts of that ecosystem. So we need to start working on developing those techniques to be more encompassing of beyond the bacteria, but also thinking more about the functionality of that bacteria as well, the different things it's able to do. It's really important to know that just because a bacteria has a certain gene, it might be dependent on the environment it's in order for that gene to do what you're expecting it to do. So there are still so many complex interactions that we are trying to understand and apply to this research. And for a long time, a lot of microbiome research was purely observational. It was so variable between populations and between subjects that it was very difficult to draw conclusions that were more than anecdotal. But that's one of the great things about looking in domestic animals such as dogs. Dogs have really similar microbiomes to each other. There is a lot less variability within the UK dog population microbiome than the UK human population microbiome. So that's one of the things that makes that dog microbiome research so valuable. And with the use of things like machine learning and AI, we're really starting to use the microbiome for actionable insights and to be able to inform clinical decision making as opposed to being purely observational.
Another huge part of your scientific journey has been the work you do in science communication and sharing your story. How did you first get engaged in the SciComm world?
I think it's something that developed quite naturally. And perhaps I was doing science communication long before I officially started doing science communication. I think because I have dyslexia, the way that I understand and process information is very big picture. And it means it makes it very easy for me to come up with analogies. And I would do this a lot when having to explain things to myself to better understand biology or certain processes that we were studying at university or at high school or sixth form or whichever. And having that ability, I've now been able to translate that into using it to explain science to even more people. Because I'm the only scientist in my family, all throughout my PhD, I spent a lot of time just doing science communication within my family, explaining why my research was important and why I was dedicating the next 5 years of my life to this. So that really was my first instance of the science communication. But also all throughout high school, I was big into performing arts. I loved drama, I loved amateur dramatics, I loved singing, I loved dancing. And I think that also helped me to develop certain communication skills. I've also done radio for a really long time. So I first started radio when I was in high school and even had my own independent radio show in Norfolk a couple of years ago. With that radio show, it was called Nature and Nonsense, and it was all about bringing listeners up to date with the latest environmental, ecological, and wildlife news. It also gave me that opportunity to explore communicating science using different formats as well. I'm a very verbal person. I'm a big storyteller, and being able to intertwine those experiences and those skills that I have with my passion for science has actually been really enjoyable for me as well. One of the things I am most proud of throughout my scientific career is my involvement with the British Science Week. So this is done by the British Science Association, and every year they have British Science Week in March, and they send these packs out to schools where the schools can then create activities around scientists and what it is to be a scientist. And I'm featured in that as part of their Smashing Stereotypes campaign. And I talk about my love for chickens as a child and my incubator in high school, and how that led to me into where I am now and led me into science, and how I always followed that passion and that curiosity. And knowing that I'm now helping to inspire some of those younger generations that perhaps came from similar backgrounds to me, or inspiring people just to follow the thing that they are most passionate about to see where it leads them, has been really fulfilling in terms of my recent career.
That's it for this episode. Thanks for tuning in. As you may have seen, we've recently launched our autumn programme of events here at the Royal Institution. So head to rigb.org to check out all of our upcoming events from September to December, and hopefully we'll see you soon.